classify epithelial tissues. Okay. The first thing we want to bear in mind is that the first name of the tissue is going to be the number of layers, right? So the first name is the number of cell layers, okay? The second name is essentially the shape of cells, specifically at the apical most layer. Right, to the top layer. So the first name is the number of cell layers, whether it's simple or stratified, and then the second name is going to be the shape of the cells that are at the very top or the apical most layer. So on that basis, we have 3 types of simple tissues, and then 3 types of stratified tissues. So the first type of simple tissue is your simple squamous tissue. Now in squamous cells, the cells are flat, just like here, and then their nucleus is also flattened right in the middle. So it's a flattened cell with a flattened nucleus, and it's 1 cell layer thick. All right, so this is your simple squamous. We're going to talk about some specific examples, so tissue examples or locations of these tissues later on, but right now we're looking at the structure. Then if we have some cube-shaped cells, which are as tall as they are wide, and it's 1 cell layer thick, this is your simple cuboidal tissue. Now, as we notice with cuboidal cells, the nucleus is not flat, but it's very round, and it's right in the middle of the cell. Okay, so it's a large, rounded, centrally placed nucleus. And then finally, columnar cells are taller than they are wide. Right, so these are your columnar cells. And the nucleus of columnar cells are stretched or ovoid shaped, and they're placed closer to the basal surface of the cell. Right, so they're closer to the bottom of the cell, and they're ovoid shaped, almost as if you took that cuboidal cell and you stretched it so that the nucleus was now elongated. Okay. So that's your simple columnar tissue. Now, columnar tissue is also associated with 2 modifications. There can be microvilli. We're going to talk about microvilli, which are these finger-like projections. Right, so these are microvilli. We'll talk about the location and function of microvilli in a minute. And then columnar cells can also be associated with cilia. And cilia are these taller, hair-like projections. So this is cilia. Okay. Now, columnar tissue is also associated with goblet cells. And goblet cells have all of these granules inside of it. This is the nucleus, which is closer to the bottom of the cell. And then all of these granules are pretty much mucus, mucin granules for mucus production. So this is a goblet cell, and its job is to produce mucus. So these are some of the modifications that are specifically associated with columnar cells. Right? We don't really see this with simple cells, with squamous cells, and we don't see it with cuboidal cells. Let's look at the stratified tissues. So the first type of tissue here, now again, stratified tissues are more than 1 cell layer thick. So the first type here is going to be stratified squamous. So we have flattened cells but we have several layers of these flattened cells. Again, their nucleus is going to be flattened, just like the cell. But at the basal layer of stratified squamous tissue, there are actually cuboidal cells down here. And these cuboidal cells are responsible for the regeneration of these flattened cells at the top. Now, because the cells at the topmost layer here are flat, this tissue is going to be called stratified squamous and not stratified cuboidal. Remember, stratified tissues are named according to the shape of the cells at the topmost layer. Okay. Next, we have stratified cuboidal tissue, which again is more than one cell layer of cuboidal cells. There are typically only 2 layers associated with stratified cuboidal, so not more than 2. And you have this large, centrally placed, very rounded nucleus. And then the last layer of stratified tissue is stratified columnar. And stratified columnar is, again, another layer of columnar cells, but underneath these columnar cells, there are actually cuboidal cells here. So there's more than one cell layer, but the apicalmost layer is columnar. So we've got these cuboidal cells that are responsible for regenerating these columnar cells at the top, which are the tissues named after these columnar cells, right? And again, we have associated cilia or microvilli, and then associated goblet cells. Alrighty, and this is your stratified columnar tissue. A very rare tissue. We'll talk about the few tissue examples in the body, but it's not very common throughout the body, stratified columnar. All right, any questions? So basically, this is how we classify tissue types in the body. We look at the cells, we count the layers, and we identify the shape of the cells at the apicalmost layer and the number of layers that are associated. Question?
Does the goblet cell have cuboidal cells?
No.
Okay.
No, the goblet cell is going to be at the topmost layer with the columnar cells. Okay. All right, any other questions? Sure. So what we're going to move on to talk about next is really how each of these tissues has a very unique function, right? And that function is going to tie very closely to the way the cells look, the shape of the nucleus, and the size of the cell. We'll see that smaller tissue or flatter cells, tissues with flat, simple squamous cells, are pretty much specialized for diffusion and filtration, so the easy movement of substances across that tissue. Whereas bigger cells, like the columnar cells, are responsible for absorption and secretion, right? And that's going to tie into the fact that a smaller cell has less space in it for machinery that is going to produce secretions, as well as the energy production to actually bring about that process of secretion and absorption. So the smaller the cell, the less machinery in the cell to bring about processes such as absorption and secretion, whereas a bigger cell has more space to, let's say, produce those secretions or to produce the energy to actually secrete those substances. Okay, done with this image. Can we move on? All right, so let's talk about the different characteristics of the cells of epithelial tissues. First of all, there's cellularity that these cells exhibit, and cellularity means that there's not a lot of stuff going on around the cells. There's very minimal extracellular material, and this tissue is mainly comprised of cells. That's what cellularity means. So when you take epithelial tissue, there's not a lot of loose connective tissue or so around it. It's pretty much cell next to cell, next to cell, next to cell, right? Specialized contacts are the structures that join cells together. We're going to talk about those. There are certain tight junctions, desmosomes, gap junctions, and so on. And they're usually at the lateral domains of the cell or the lateral surfaces of the cell. And we'll talk more about the unique functions of each of those and where they're located along the lateral domain. Then polarity. Polarity is the idea that cells are not the same on all sides. So the apical surface of the cell, which is the surface that projects upward or inward. So let's take this tissue, for example. The apical surface would be facing the lumen. So let's say we took the gastrointestinal tract. Let's say we cut it in two so that we got a cross-sectional surface. The cells or the surface of the cell that faces the lumen is the apical surface. And the apical surface has different characteristics and different properties than, say, the lateral surface or even the basal surface. So polarity basically means that each surface of the cell differs greatly in the way it looks and the way it functions. Connective tissue is basically the supporting tissue that is underneath the cells. So for example, this is your epithelial layer, which is formed by several columnar cells in this example. And then the tissue underneath it is basically providing nutrients and anchoring it down to the underlying tissue. This brings about the point that the epithelial tissues are avascular, meaning that they do not have an active blood supply. They have to get blood, they have to get their oxygens and nutrient diffused from the underlying capillaries. Okay? So the blood vessels that are in the connective tissue will diffuse oxygen and nutrients from the blood into that epithelial layer because these blood vessels do not actually go into the epithelial tissue. But they are innervated. So as you can see, the nerve endings do actually leave the connective tissue and go up into the epithelial tissue. So they are avascular, meaning no blood supply, but they are innervated. So they do receive those nerve endings, and they do have a sensory component. Finally, the idea that regeneration of epithelial tissues always takes place from the basal layer upwards. So this is specific for stratified tissues where you have a cuboidal layer of cells at the bottom and let's say a stratified layer of cells at the top. The regeneration takes place from the basal layer. That's because this layer is closest to the blood supply and takes place in an upward direction to the apical layer. So these are some characteristics and properties of epithelial tissue. The most important ones are the polarity. So there's an apical, basal, and lateral domain to the cells, as well as the idea that epithelial tissues are avascular but innervated.